Okay, so welcome back from your break, whether you shut this down, paused it, and took a little bit of a break, or you just clicked to come on to the next portion of our discussion. Let's take a look at the concept check questions and get those answered so we can continue our look at meiosis. So how does an asexually reproducing organism produce offspring that are genetically identical to each other and to the parent? Well, the clue, of course, is the offspring are genetically identical to each other and to the parent. That is, of course, our definition of the purpose of mitosis. Mitosis produces cells that are genetically identical to each other and to the parental cell. So if you are an asexually reproducing organism and this is your goal, then you're going to utilize mitosis. So that would be the answer here. You want to produce many copies of a very popular new orchid. What should you do? Breed it with another identical orchid or clone it? Well, if you breed it with another identical orchid, that means you're going to be using sexual reproduction. And as we have been talking about in the previous section, the whole purpose behind sexual reproduction is to create genetic diversity. So if you were to breed the orchids, what you would wind up with is orchids that are likely not exactly the same as the orchids that you started with. If you want them to be identical, then the only way that you can guarantee that is to clone it. And in fact, that is the way the vast majority of orchids are produced these days. They are actually clones. Not because people want to necessarily want to guarantee that the clones are identical to the starting orchid and therefore all the orchids are identical, but because if you do try to sexually reproduce orchids, in other words, you crossbreed orchids, you're going to have a very, very low success rate. It turns out that the success rate of germinating orchid seeds is about 1 in 100 seeds. So out of every 100 seeds that you try and germinate, only about one of them is actually going to germinate and give you a new orchid. But if you're trying to grow orchids for commercial purposes because you want to sell them, that's really not going to cut it because you're not going to be able to produce enough orchids. So orchids are actually cloned, and it's relatively easy to do it. When I taught plant physiology over at UCF, which I used to do when I was on the faculty over there, we used to do a plant cloning experiment every semester. And we would start on week 1, and by the end of the semester, students were taking home plants that they could plant out in their garden. And I would let them do that as long as they bought seeds back once those plants produced seeds so that I could grow more plants for doing more cloning because it was a source of seeds. We actually used to use tobacco plants simply because tobacco plants are a type of plant that we work a lot with in the laboratory because they're easy to grow and easy to manipulate. And it's very easy to clone them. But orchids commercially are cloned. So this is what the best way to guarantee that you're going to have lots of orchids that are identical to each other. And so from a single orchid leaf, you might be able to produce conservatively maybe 500 to 1,000 clones. I know that seems like a lot, but the nature of how you go about cloning plants means that you can generate large numbers of plants from a very small amount of leaf tissue, and primarily we do the cloning off leaf tissue. It's an interesting topic. I wish we had more time to talk about it, but we don't. We're talking about meiosis here. So let's move on and start talking about meiosis. So before the break, I went to a lot of trouble to emphasize that the whole point behind meiosis is to reduce the number of chromosomes from the diploid number to the haploid number. In humans, the diploid number is 46, but remember that means 2 complete sets of chromosomes. So 1 complete set of chromosomes is not 46. 1 complete set of chromosomes is 23 chromosomes. So Let me just put that in here. So one complete set of chromosomes is 23 chromosomes for a human. Now, we will use human numbers a little bit as we go through this chapter, simply because it's what most people are familiar with and it allows us to illustrate the concept. But one of the things that I strongly encourage you to do is to get out of the habit of thinking 46 and 23 for diploid and haploid. That's only true for humans. It's not true for other species. And if I talk about other organisms or ask exam questions about other organisms that have different numbers of chromosomes, you may or may not be able to answer the question if all you know is diploid 46, haploid 23, the human condition. What I would prefer you to get into the habit of thinking about is diploid and haploid 2N and N. In other words, the general idea. So let me put this in down here. So diploid is designated 2N. Haploid is designated N. So again, you can see in our mathematical representation, we're using the concept of 2 complete sets and 1 complete set, 2 times N and N. So the way we can demonstrate that this is the case is to use a technique called a karyotype. This is not something that is done routinely. It is typically done when there is some suspicion of a genetic abnormality. And the only way that you can really see that genetic abnormality at the chromosomal level is to do a karyotype. So we typically will do this on a cell that is undergoing meiosis. The reason why is because during meiosis, as you'll see, the chromosomes that constitute the 2 sets appear paired up with their partner chromosome. And so when we take a look at a karyotype of a human, this is what we see. So here is a karyotype of an individual. I happen to be able to tell you that this individual is male because of a particular pair of chromosomes. So let me change to red. The pair of chromosomes that are telling me that is this pair. And this pair are referred to as the sex chromosomes. In this case, male. The other pairs of chromosomes, the other 22 pairs of chromosomes that humans possess, those are the autosomal chromosomes. So let me put that in here as well. So you can see that we have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of gender chromosomes or sex chromosomes. Now, if this was a female, she would have 2 copies of the X chromosome, and there would be 23 pairs of identical chromosomes. For the purposes of I'm talking about meiosis, for the purposes of talking about meiosis as a form of cell division and identifying the pairs of chromosomes, we do consider the X and Y chromosomes to be a pair of chromosomes, what is known as a homologous pair of chromosomes. Because during meiosis, as you'll see, the homologous pairs of chromosomes do pair up. In other words, each chromosome pairs up with its identical partner, and in a male, the X and the Y pair up during meiosis. So we do consider them to be a pair of homologous chromosomes, even though they're not identical to each other, which is captured in the term homologous. Remember, the prefix homo refers to the same. And so a homologous pair of chromosomes are chromosomes that are the same. So what do we mean by homologous? What do we mean by the same? Well, if you look at the artist's rendition of a pair of homologous chromosomes in the center of this slide, you can see number one, they are the same size. In other words, what are referred to as the two arms, represented by this part of the chromosome and this part of the chromosome. So there are the two arms, and the two arms are what extends out from either side of the centromere. The two arms in each chromosome are the same length, so they're the same size. The position of the centromere within the chromosome is identical. If you stain the chromosomes with colored dyes, then what you notice is that the dye pattern is exactly the same in the two homologous chromosomes. So they're identical in size, they're identical in shape, they're identical in terms of how they take up colored dyes. If we go to the genetic level, oops, I won't go there just yet. If we go down to the genetic level, and here I'm going to have to draw a diagram to illustrate this. If we think of these as the two homologous chromosomes, then if we look on the chromosome at a particular location, and we find that there is a particular gene for eye color at this location, we're going to find a copy of the same gene in the same location on the homologous partner. If we go to another location and we find that there's a gene for hair shape, at that particular location, in the same location on the homologous partner, there'll be the identical gene for hair shape. I could throw a third gene on here, but I think you're beginning to get the idea. If we look at a particular gene on a particular chromosome and find a particular gene in that location, when we look on the homologous chromosome in the same location, we're going to find the same gene. So there's one other term that I want to introduce here. I think I can fit it in here. So a gene's location on a chromosome is its locus. And the plural is loci. So when we talk about the eye color gene, when we talk about its physical position on the chromosome, we are talking about the locus or locus of that particular gene. If we look in the same location, in the same locus or locus, depending on how you want to pronounce it, on the homologous partner, you're going to find the same eye color gene at that same physical location, at that same locus. So this is all part of our understanding of what makes two chromosomes homologous. They're the same size, they have the same shape, same location of the centromere, they take up colored dyes in the same pattern, they have the same genes in the same physical locations, the same loci. There is, however, one subtle difference, and that is the version or flavor of the gene. So I don't have room to put that on this slide, so I'll put it on the next slide because it might be appropriate to talk about it there. So let me put this in here. So homologous chromosomes. Can differ in the version or flavor. So let me just apply the eraser there. That's where I should have gone. These different versions are called alleles. So how can we illustrate this? Well, if you think about eye color, and if we assume that eye color is the product of a single gene, it's not, but for the purposes of the argument, we'll think of it as being the product of a single gene. Then you know that there are different eye colors. So there's blue, brown, hazel, green. There are multiple different eye colors. And it's quite possible that your parents have different eye colors from you. But certainly they may have different eye colors from each other. Now, you may have the same eye color as one of your parents. You may have a different eye color from both of them. The point is, we know that your parents made contributions, genetic contributions that did ultimately lead to your eye color. So if their eye colors are different from yours and different from each other's, how do we account for that? Well, if we assume that, again, that eye color is the product of a single gene, the only way we can account for that is if there are different versions of that eye color gene. So there must be a blue version of the eye color gene. There must be a brown version of the eye color gene. There must be a green version of the eye color gene, a hazel version of the eye color gene. Each of those different versions or flavors is known as an allele. So where there is a blue allele of the eye color gene, there is a green allele of the eye color gene, a brown allele of the eye color gene, a hazel allele of the eye color gene, and so forth. So we also have to think in terms of two other relationships that come up in discussing genetics and inheritance. And that is the notion of characters and traits. Now, this is not so important in this chapter. It's more important in chapters 14 and 15. But I'll introduce you to the concept because we do have to have the notion of genes and their alleles in this chapter to understand the source of genetic variation a little bit later in the chapter. But we can also relate characters and traits to genes and alleles. So when we talk about eye color, we're talking about a character. When we talk about hair shape, we're talking about a character. When we're talking about, a hitchhiker's thumb versus not a hitchhiker's thumb, we're talking about a character, the particular version of that character that you have. So blue eyes, or a hitchhiker's thumb, or curly hair, those are traits of the particular character. So it's the blue-eyed trait of the eye color character, the curly-haired trait of the hair-shaped character, the hitchhiker thumb trait of the thumb-shaped character. And so when we start thinking about that, it becomes very obvious that a character is the product of a gene. The particular version of the character that you have, the trait, must be the product of the particular version of the gene you have, or to use the proper term, the allele. So if you have the blue-eyed trait of the eye color character, that means you must also have at least the blue allele of the eye color gene. Hopefully you understand that connection. If you don't, we will come back and be talking about that again in chapters 14 and 15, particularly chapter 14. So if that doesn't make sense right now, that's okay. It's not terribly important in this chapter, but the concept of gene and allele, that most definitely is. Before we finish this chapter, you will have to understand the concept of gene and allele because it is a source of genetic variation. And remember, that's what this chapter is all about. So I've shown you the presence of the X and the Y chromosome in that karyotype. It indicates male, and I've already told you that XX gives you female. So we know what the chromosomal makeup of any somatic cell is in an individual. What we need to bear in mind is that the germ cells in the testicles and the ovaries, if we're still talking humans and we're still talking animals, which we are at the moment, then the germ cells that are going to undergo meiosis are, to all intents and purposes, somatic cells, because that's how they start out their life. They start out chromosomally identical to somatic cells. They just happen to be located in the testicles, in the ovary, and they are destined to undergo meiosis. So they start out with 2 complete sets of chromosomes. So in humans, they have the 46 chromosomes to 23 pairs of chromosomes. But there's an added twist that we have to add on top of this. So just to remind ourselves again, the human life cycle, you're familiar with this. I don't need to talk about this in detail. It's the animal life cycle. You know how this works. The added twist to all of this is that this is still cell division. And hopefully you recall the 3 steps of cell division from mitosis. The fact that before cell division begins, you've got to duplicate the chromosomes. Then during the division process, you've got to separate the copies of the chromosomes, and then ultimately you can divide the cell into daughter cells. Think about that for a minute. We are trying to create gametes that have 1 complete set of chromosomes. Fair enough. But we're starting with cells that contain duplicated chromosomes. I mean, that's the end product of duplicating the DNA. We learned that in the last chapter. So even though we are trying to halve the number of chromosomes, we still have to take into account the fact that those chromosomes are going to contain 2 sister chromatids, and we know that eventually those 2 sister chromatids are going to have to be separated, and the destiny of those separated sister chromatids is to become chromosomes. Which, if we're talking about human cells, means that we're not dealing with 46 potential chromosomes in the end, we're dealing with 92 potential chromosomes. Because if each chromosome, 46 of them, gets duplicated before we begin, which they do, and each contain 2 sister chromosomes as a consequence of that, which they do, then it's 46 times 2 chromatids, which means 92 chromatids, and those 92 chromatids represent 92 future chromosomes. That's the extra twist that we have to take into account here. And so what it means is that meiosis cannot simply be a single division of the cell, because that wouldn't do it. It might reduce the number of chromosomes in half, but we'd still be stuck with chromosomes containing 2 sister chromatids, the duplicated chromosomes. And that can't be allowed to continue because that gives you too many copies of each chromosome. So consequently, when we start considering meiosis, we have to recognize that meiosis must be, of necessity, a two-phase or two-stage cell division. One stage involves the reduction of the chromosome number from diploid to haploid, from 2N to N. But the other division has to take into account the fact that each of those chromosomes contains two sister chromatids and must somehow separate the two sister chromatids. But wait a moment. We already know how to separate two sister chromatids. We saw that last week, called mitosis. So as much as we need meiosis to create gametes, we're not completely divorced from mitosis, because it turns out that the second stage of meiosis, what we refer to as meiosis 2, to all intents and purposes, is mitosis. It certainly looks very much mitosis, like mitosis. There are some subtle differences, as you will see. And one of our concept check questions is asking about the differences. So something to pay attention to. But nonetheless, when we look at meiosis 2, which is all about separating sister chromatids, it's really just a replay of mitosis. The stages look exactly like the stages of mitosis. So in reality, we already know half of meiosis. We know the second half. What we really need to focus on is the first half, where things are going to be a little bit different from mitosis, because they have to be, because we're not trying to create genetically identical cells. We're trying to create cells that contain one complete set of chromosomes rather than two complete sets of chromosomes. We want to go from diploid to haploid. We want to halve the number of chromosomes. That's something that mitosis can't do, nor will it ever be able to do. So meiosis 1 is where all the differences between mitosis and meiosis actually exist. And that's why we spend more of our time focusing on meiosis 1 and coming to terms with that. Can't ignore meiosis 2, because it is still part of meiosis and it's a very important part. But we know what it consists of, because it's trying to do exactly the same thing as mitosis, separate sister chromatids. So to all intents and purposes, it looks just like mitosis. So this is what we start with. You can see here a cell that has 6 chromosomes in total. That means its diploid number is 6. 2n equals 6. But you can also see here that each of the chromosomes has been duplicated. So each chromosome contains 2 sister chromatids. So the reality is there are 12 DNA molecules here, 12 potential chromosomes or future chromosomes in the form of those sister chromatids in each of the 6 chromosomes. 6 times 2 is 12. Now notice we're throwing in, or we're introducing, a new term, non-sister chromatids. Non-sister chromatids are chromatids that are not in the same chromosome. What they are sister chromatids in each of the 2 homologous chromosomes that make up a homologous pair. So you can see in this pair of homologous chromosomes, this is one sister chromatid, and this is the other non-sister chromatid. They don't exist in the same chromosome, duplicated chromosome. One is in each of the 2 homologous chromosomes that make up the homologous pair. So why introduce this terminology? Because it's going to become really important. Because we're going to create a connection between these 2 non-sister chromatids in order to keep the 2 homologous chromosomes linked together during the early part of meiosis, and specifically during the early part of meiosis 1, so that the pairs of homologous chromosomes are moved around together. For the same reason we saw the 2 sister chromatids linked together by the centromere during mitosis so that they could be moved around together as a pair during the initial stages of mitosis until it was time to separate them. So we want to move the two homologous chromosomes as a pair together until it's time to separate them at some point during meiosis 1. And the way we keep them together is to create connections between the non-sister chromatids, what are called synaptonemal complexes. Really big term, but it basically means very real physical connections, protein connections, between the two non-sister chromatids to hold them together until such times it's time to separate them at some point during meiosis 1, as we'll see. So this is what we start out with. So time for another break before we go on and actually talk about the nuts and bolts of meiosis, because that's what the next stage is all about. So in the cell shown in slide 23, this one, how many individual DNA molecules or potential chromosomes are there? Something that I talked about. What is the haploid number, and is it a set of chromosomes, and is a set of chromosomes diploid or haploid? Something to think about. In a human karyotype, how many pairs of chromosomes are present? How many sets of chromosomes are there? I'll see you on the other side of the break with the answers.